In this video, we will look at concept of homeostasis and regulation of blood glucose level. Homeostasis refers to the physiological process where organisms maintain constant and balanced internal environment despite the changes in external environment. There are three components in homeostasis, which are receptor, control center, and effector. Receptors are any cells that detect stimulus, which is changes from the normal environment. Example of stimulus is increase in body temperature. Receptor will then send input to control center. The control center will evaluate the input from receptor and provide output to effector. The output is in the form of electrical impulse, hormones, or other chemical signal. The effector will receive the output from the control center and will produce response to correct the stimulus. There are two mechanisms in which the effector will provide response, either by negative feedback mechanism or positive feedback mechanism. In negative feedback mechanism, the response will reduce the intensity of the original stimulus, where it causes changes that is in opposite direction from the stimulus. For example, when body temperature increases, our body will sweat. This is a cooling mechanism to decrease the body temperature and return the body temperature to normal. As you can see, the negative feedback mechanism responds in opposite direction from the stimulus. Most of the process in homeostasis will involve negative feedback mechanism, such as regulation of body temperature and regulation of blood glucose level. In positive feedback mechanism, the response is to increase the intensity of the original stimulus. For example, during parturition, the uterus contraction will result in secretion of hormone oxytocin, which will further increase the intensity of the uterus contraction. Next, we will look at the regulation of blood glucose level, which are controlled by negative feedback mechanism. The regulation of blood glucose level involves alpha cell and beta cell of islet of Langerhans in the pancreas. Islet of Langerhans will act as receptor and control center in the regulation of blood glucose level. During hyperglycemia, where blood glucose level is higher than normal, beta cell of islet of Langerhans will detect these changes and secrete hormone insulin. Insulin is then distributed to all cells, especially liver, muscles, and adipose tissue to increase the uptake of glucose from the blood. These tissue act as the effector. Insulin will promote glycogenesis in the liver, where this is a process that converts glucose into glycogen. Insulin will also promote cellular respiration in liver and muscle tissue, where glucose is used to generate ATP. Next, insulin promotes the conversion of glucose into fats. The uptake of glucose from blood by liver, muscles, and adipose tissue will cause the blood glucose level to decrease. The presence of insulin in the tissue will also inhibit glycogenolysis and gluconeogenesis, both of which will increase blood glucose level. Since these two processes are inhibited, blood glucose level will return to normal. Condition where blood glucose level is lower than normal is known as hypoglycemia. Alpha cell will detect these changes and secrete hormone glucagon. glucagon will be secreted to all tissue. This hormone will promote glycogenolysis in liver and muscle tissue, which convert glycogen into glucose. Glucagon will also promote gluconeogenesis in adipose tissue, where fat is hydrolyzed to glycerol and fatty acid and then is converted to glucose. Glucose from both glycogenolysis and gluconeogenesis will enter the blood and increase the blood glucose level. In addition, other hormones such as growth hormone and adrenaline will also raise the blood glucose level. Glucagon will inhibit glycogenesis, which is the conversion of glucose to glycogen, so that glucose remains in the blood. All these processes will increase blood glucose level and return it to normal. Okay, so that is concept of homeostasis and regulation of blood glucose level.